respiration is of two types. The respiration is either anaerobic or aerobic. Anaerobic or aerobic. When it takes place in the presence of oxygen, we call anaerobic. When in the absence of oxygen, oxygen is not there, we call it anaerobic. And then we did that. What is the, let's start with this. It will be a fast recap of whatever we have done. In anaerobic respiration, the one thing before I start with anaerobic, I want to tell you that glycolysis, which is the first step of respiration, is common in both types, whether it is aerobic or anaerobic. This is common in both types. It takes place in both types, and the site of glycolysis is cytoplasm. Where it takes place is cytoplasm. Now the anaerobic respiration, it goes on like this. We have glucose as a source of energy. The glucose undergoes glycolysis in the cytoplasm. Glycolysis. Glycolysis means breakdown of glucose. It takes place in the cytoplasm, and when it takes place in the cytoplasm, this releases 2-pyruvic acid. So pyruvic acid is formed, which is 2 molecules, and then these 2 molecules, they, in case of anaerobic respiration, which takes place either in yeast or in the muscles, either in yeast or in the muscles. What happens in the yeast? In the yeast, CO2 is given out. After the giving out of CO2, a 2-carbon compound with the C2H5OH is formed. Now here we have 6, because pyruvic acid is a 3-carbon compound and we have 2 pyruvic acids, so 6 carbons are there. And here we have this 2, okay. Here this is a 2-carbon compound, C2H5OH. So 2 ethanol is formed here, and 2 carbon dioxide are released. So it completes now. See, it is 4 and it is here 2, 6. So 6 are done. So ethanol is produced, and what happens in the muscles? In muscles, lactic acid is produced. Lactic acid, the formula of lactic acid, which I told you, C3H6O3. This is produced. Okay, and in both the type, 2 ATPs are released. 2 ATPs are released, 2 ATPs are released, and 2 lactic acids are produced. So when 2 lactic acids are released, that means 6, and it is also 6, because this is a 3-carbon compound and 2 are produced, so 6. So this is 6, the 6 carbon are done. So this is what happens in anaerobic respiration. When we come to aerobic respiration, in aerobic respiration, what happens here? The glucose, this glucose undergoes the process of glycolysis. This is the layout which I am telling you. Then this takes place in cytoplasm, as you all know. And after this glycolysis, what takes place is Krebs cycle. And this Krebs cycle takes place in mitochondria. And this what, see, as a result of this, what is produced here? As a result of this, the pyruvic acid is produced, which through gateway step, here one gateway step is also there, which is oxidative decarboxylation takes place, and this pyruvic acid gets converted into acetyl coenzyme A, and then which enters the Krebs cycle. After Krebs cycle, it enters the electron transport chain, where the NADH2, which is produced, is, you know, it splits and results in the formation of water. And at the same time, this takes place in inner membrane of mitochondria. Inner membrane of mitochondria. And this takes place, then it is oxidative phosphorylation. And this oxidative phosphorylation, this takes place in F1 particle, and results in the synthesis of ATP. So ATP is produced here, and this is the layout of the aerobic respiration. And you asked me a question that day. I was asked a question by you all that what you are going to write, whether it is 36 ATP or 38 ATP. As I told you, 2 ATPs are used up in bringing the pyruvic acid from the cytoplasm into the Krebs cycle via oxidative decarboxylation. Okay, now that is also used up. So if it is an objective type question and you are given just both the options, either 36 or 38, you're going to write 36. You will write 36. 36 ATPs are produced. Don't mention 38. And if it is 1 mark or 2 marks question, then in that case, we are going to explain that in all 38 are produced, and out of those 38, 2 are utilized in transferring the pyruvic acid from cytoplasm to the mitochondria through oxidative carboxylation step. So 2 ATPs are used there. That is why the net production is 36 ATP you are going to mention. But if it is objective type, it's okay to write 36 ATP. Otherwise, you mention 38 and then write why 36 are left at the end of the aerobic respiration. Now, beta, anybody who remembers what is the other name for glycolysis? One more name is there of glycolysis. Anybody who remembers what is the other name for glycolysis? Yes, anybody who remembers the other name for glycolysis? Yes, bache. Anybody who remembers the other name of glycolysis? I'm not getting any response from anybody. These days, you people have stopped responding to that means you are not preparing your topics which you study in the class. Yes, beta. Amrita is saying EMP. Yes, it is EMP. Can you tell me the full form of glycolysis? The full form of glycolysis is Emden-Meyerhof. Yes, EMP pathway. But what is the full form? Emden-Meyerhof-Parnas. Yes, Emden-Meyerhof-Parnas. Now, what are these names? Emden-Meyerhof-Parnas. What are these? Can you tell me what are these? Emden-Meyerhof-Parnas. What are these words? Yes. Can you tell me what are these words? These are, yes, these are the people who discovered this pathway. These are the researchers, the scientists who did work on this, and on their names, this name is given EMP pathway or Emden-Meyerhof-Parnas pathway. This pathway is known as glycolysis, in which glucose, which is the source of energy, which is the food, ultimate food in a body, that is broken down to give pyruvic acid. And how many molecules of pyruvic acid are formed? It's two. Two pyruvic acids are formed. Now here, what happens? Here, in case of glycolysis, this is also known as EMP pathway. Also known as EMP pathway, and this takes place in both anaerobic and aerobic, anaerobic as well as aerobic, and aerobic respiration. In both the types of respiration this takes place, the site is cytoplasm. It takes place in cytoplasm. Okay, then see, in plants what happens? Sucrose is there. Now this sucrose, see, in animals, fine, we have glucose in us. In animals, in the yes, in animals we'll call, in animals glucose is there. We have glucose to start with this, with the process of the glycolysis. But in plants, what do we have? In plants, we have in plants they have sucrose. And this sucrose is further broken down into fructose and glucose. And this glucose, it undergoes the process of glycolysis. Now, what is the process of glycolysis which takes place? The glucose utilizes ATP. This is what we have. Glucose utilizing ATP, when ATP is utilized, what happens? ATP gets converted into ADP. This is utilized, gets converted into glucose 6 phosphate in the presence of enzyme hexokinase. Okay, glucose 6 phosphate. This glucose 6 phosphate, under the action of another enzyme, which is isomerase, gets converted into its isomeric form, which is fructose 6 phosphate. This fructose 6 phosphate again utilizes one ATP here. One ATP is utilized. So ATP converted into ADP. And here this gets converted into fructose 1,6 biphosphate. Fructose 1,6 biphosphate. This then splits, and on splitting it forms dihydroxy acetone. Dihydroxy acetone. And also glyceraldehyde 3 phosphate. This is formed. Both are 3 carbon compounds. And then finally what happens after this? What happens that this here one NADH2 is formed. NAD gets converted into NADH2. This is formed, and this then forms 1,3 biphospho glycerate or glyceric acid. Then this 1,3 biphosphoglyceric acid, here one ATP is synthesized. Here they are used, here they are synthesized. Here these two molecules of this, and then here ADP plus P gets converted into ATP. Two of these ATPs are formed. Then 1,3 biphosphoglyceric acids get converted into 3 phosphoglyceric acid, and then this 3 phosphoglyceric acid gets converted into 2 phosphoglyceric acid. This 2 phosphoglyceric acid under the action of enolase gets converted into phosphopyruvate. Phosphoenol pyruvate. And here what happens? Again, one NAD gets converted into NADH2. Here? Not NADH2. I'm writing wrong here. This is ATP. ADP forms ATP. ADP plus P forms ATP. Again, 2 molecules of these are formed. So 2 of this, 2 of that, and finally we get pyruvic acid. Pyruvic acid is a 3 carbon compound, and 2 molecules of these are formed. Pyruvic acid. That means how many carbons we have? It's a 3 carbon compound, so 2 are formed. That means 6 of the carbons, and we'll have to utilize 6 of the carbons in Krebs cycle and oxidative decarboxylation. Now here, if we see the ATPs formed here 2 ATPs are formed, here 2 ATPs are formed. So ATPs formed are 2. 2 here, 2 here, 4. So 4 ATPs formed. ATPs utilized. ATPs utilized are 2, so minus 2. So we get 2 here. Out of the ATPs, we get 2 here. But 2 NADH2 are also formed. So 1 NADH2 gives you 3 ATPs. So 2 NADH2 will give 6 ATPs. So how many are formed? Here 2 are formed, so 6 ATPs. So 6 ATPs are formed here, 2 of this. So 6 plus 2 is equal to 8 ATPs. 8 ATPs are formed in the process of glycolysis. So in all, during the process of glycolysis, 8 ATPs are formed. So this was glycolysis. Then after glycolysis, the pyruvic acid molecules enter the process of oxidative decarboxylation. So the next is oxidative decarboxylation, also known as gateway step. So oxidative decarboxylation takes place. Now this oxidative decarboxylation is a uh, step which utilizes magnesium. Magnesium is utilized, and along with magnesium, an enzyme, pyruvate dehydrogenase. Dehydrogenase. This enzyme is very important, which is utilized in the gateway step. And because of this gateway step, what happens? The pyruvic acid, which are two molecules, from them, the C coenzyme A is added. Coenzyme A is added and CO2 is removed. Decarboxylation. So CO2 is removed, and along with this, what is formed is NADH2. NAD gets converted into NADH2. This is converted into this, and finally results in the synthesis of acetyl coenzyme A. Acetyl coenzyme A is formed. Now this acetyl coenzyme A, this enters the Krebs cycle. This will enter the Krebs cycle. Excuse me. So now this, after this, this acetyl coenzyme A enters the Krebs cycle. When it enters the Krebs cycle, first of all, this is received by acetyl oxaloacetic acid. So OAA receives it, and after receiving oxaloacetic acid in the presence of another enzyme, which is citrate synthetase. Citrate synthetase, citric acid is formed, and this citric acid is formed. After the formation of citric acid, its isomer is formed, which is isocitric acid. Isomer of citric acid, which is isocitric acid, and here isomerase enzyme is used. Here citrate synthetase is used. Then after this, what happens during the process? NAD gets converted into the high energy compound NADH2 with the release of CO2 here. CO2 is released, it forms NADH2, two molecules of this is formed and gets converted into alpha ketoglutaric acid. It gets converted into alpha ketoglutaric acid. Now, to this, what happens again? One NAD is produced, NADH2 is produced here. Coenzyme A is added. Here, CO2 is given out. Okay, and succinyl coenzyme A is formed. Succinyl coenzyme A is formed. This succinyl coenzyme A again, from this CO2 which was added, it was given out, COA. Okay, and from GDP, guanosine diphosphate, guanosine triphosphate is formed. So, and one guanosine triphosphate, it gives one ATP only. It is like ATP only. Whereas this gives 3 ATPs. It is like ATP only, so one GTP on lysis gives 1 ATP. Okay, then it forms succinic acid. This succinic acid then synthesizes FED. One FAD is formed. Flavin, same. It is like the NADH2, and the full form of this is flavin adenine dinucleotide phosphate. Adenine dinucleotide, no Fe is there, so dinucleotide only. So FED gets converted into FADH2. This on hydrolysis gives 2 ATPs. This gives 2 ATP. This on breakdown gives 3 ATPs. This on breakdown gives 1 ATP. So fumarate, this results in the formation of fumarate. Then this fumarate gives you malate, and the malate then forms oxaloacetic acid by forming 1 NADH2. This was formed. So 2 molecules of this, 2 of this, 2 of this, 2 of this, 2 of this are formed. So this was the Krebs cycle which took place. Then we had the electron transport chain in which this NADH2 gets split, and on splitting it forms NAD. When it splits, it forms NAD, then hydrogen ion and electron. The electron passes through the inner membrane of mitochondria from one complex to another complex, and this gets deposited in the intermembrane space of mitochondria. What is that intermembrane space of mitochondria? Like after Krebs cycle, ETS is there. Okay, I want to tell you one thing, that tomorrow I can ask any 2 students to speak on any topic of respiration. It can be glycolysis, it can be oxidative decarboxylation, it can be Krebs cycle, it can be electron transport chain, it can be amphibolic pathway, or it can be the respiratory quotient. Out of these topics, I'll ask any 2 to 3 students to speak out. So tomorrow, you all will be unmute and just be prepared with any of the topic. You can give me your topic. Amrita, you can prepare one. Tadarani, you can prepare one. Yashika, you can prepare one. Then Sanvi Rana can prepare one. Harsha Sanil can prepare one. So all of you will prepare at least one topic, and I'll ask 3 to 4 students to speak on those particular topics. Okay? Okay? Yes. So tomorrow will be your session. Tomorrow will be your session. I would like to know whether you people are doing it. You are on your own also, something you are revising it or not. So tomorrow will be your session. Yashika, Amrita, Sanvi, Tadar, Priya can also be there. Then all of you take one topic. Fine. Priya is also there. Then yes, the boy, what's his name? I forgot. The boy from Jammu, what's his name? Yes, the boy from Jammu, what's his name? Rohan Wazir. Yes, Rohan Wazir, you are also going to prepare one topic. You with which topic you are comfortable, Yashika? Yashika, which topic? You are comfortable with which topic? Yashika Bhardwaj. Glycolysis is okay for you? Yes, Yashika. Along with the balance sheet, it should be. Okay. Yashika takes glycolysis. Then Amrita. The student who will take Krebs cycle will have to take oxidative decarboxylation also, because that is just single step. So oxidative decarboxylation and Krebs cycle. Amrita, is that okay? Yes, Amrita, is that okay? Aerobic respiration complete? Okay, fine. So Amrita will take oxidative decarboxylation and the topic Krebs cycle. Then ETS, who's going to take ETS? Yes. Tadarani, ETS is okay? Tadarani, ETS, electron transport chain? Okay, fine. Tadarani. Then Sanvi is there. Sanvi, the next topic, which is oxidative phosphorylation. Sanvi Rana, oxidative phosphorylation is okay? Okay, fine. Sanvi is saying yes. Then we have Priya. Priya, are you there? Ananya Gupta is also there. Ananya Gupta. Yes, Priya is also there. So Priya, which topic you are going to take? Ananya and Priya. Out of you, Ananya, you can take respiratory quotient, and Priya, you can take amphibolic pathway. Ananya, respiration, respiratory quotient. Priya, amphibolic pathway. Fine. So tomorrow you be prepared. Priya, yours is amphibolic pathway. Ananya Gupta, yours is uh, respiratory quotient. So tomorrow be prepared with your topics, and I'll ask you to deliver a short lecture which will be based on your topic. Fine. We'll record it and we'll send it in the group. Fine. So beta, see, no ATP is synthesized in ETS. You know, ETS just is the splitting of NADH2, so that, like, this is the mitochondria we have, and in the mitochondria we have this inner membrane which is folded in cristae like this. So here this is intermembrane space. Intermembrane, always inter means in between. So intermembrane space, this is the space in between the two membranes. So inter means in between. This is inner membrane, this is outer membrane. Okay, so in the inner membrane, what do we have? In the inner membrane, what takes place in this membrane is this inner membrane, NADH2 gets broken down to form NAD plus hydrogen ion plus electrons. Okay, no, this is not here, this is in the matrix, not here, not here, this is in the matrix. Now this takes place, this is the matrix, this takes place in the matrix. Now here I'll make the inner wall and this is outer. So what happens in the inner? Now what happens in the inner membrane? Inner membrane has complexes. So in these complexes the electron passes, the electron passes in these complexes. This is what takes place in the inner membrane. Now because of this, you know, the energy is released, and that release of energy shifts this, the proton, from the matrix into the intermembrane space. So this is the intermembrane space. Here the protons increase in number. Here the protons increase in number, and we have our that F0 F1 particle on the inner membrane only. Here we have, and then when this proton gradient is broken, what happens? Because we have that F0 F1 here, so energy is released, and that released energy helps in the synthesis of ATP by the process of oxidative phosphorylation. What is formed is ATP. This ATP is synthesized between F0 F1 particle which is placed on this, and the proton gradient develops more of 2H, many 2H gets accumulated here. So proton number high here and low here. So when this is broken, it passes through this F0 F1 particle which is placed here, and proton gradient is broken with the release of energy, and that energy is utilized in adding up ADP and P and forming ATP. This is what happens. Now, when we see the balance sheet, in the balance sheet, I told you 38 ATPs are formed, and out of those 38 ATPs, 2 are utilized for transferring the pyruvic acid from cytoplasm to the mitochondria through the oxidative decarboxylation. Now, when we see the balance sheet, in the balance sheet we have seen there are three processes which we will take. One is glycolysis, another is oxidative decarboxylation, and the third process which we are going to take is Krebs cycle. These three processes we will take in the balance sheet. So if we see the balance sheet, the ATPs, 2 ATPs are produced here. Why 2 ATPs? Because actually 4 were produced. 4 ATPs were synthesized These were synthesized, and out of these, two ATPs were utilized. So because of utilization of this, we got only two ATPs. So ATP is 2, and NADH2, rather NADH2, how many were produced? 2. So ultimately it gives 6 ATPs. So in all we got 6 plus 2, 8 ATPs were produced here. 8 ATPs were produced in the glycolysis. And in case of oxidative decarboxylation, 2 NADH2 were produced. Here in this, 2 NADH2 were produced. So they yield you 2 threes are, because 1 NADH2 is 3 ATP. So 2 into 3, it gives you 6 ATP here. And what about this one, the Krebs cycle? Krebs cycle, we have, okay. In the Krebs cycle, we have 1 GTP, 2 GTP is produced, 2 GTP. So 2 GTP is equal to 1 ATP. And besides this, 3 NADH2 were produced in all 2, 2, 2. 3 times these were produced, and each time 2. So we had the number of ATPs, 6 NADH2 were produced. So 6 into 3, because 1 yields, so 18 ATPs were produced as a result of the formation of NADH2. Then 2 FADH2 were also produced. These FADH2 yield you 2 into 2 is equal to 4. So as a result of this, we got 12 ATPs. Okay, 12 ATP from one pyruvic acid.